Good morning children, welcome to AIMS India's online classes. This is biology session. You are watching biological classification part 1. Children, you know very well in this world there are many living organisms. In order to study them systematically, in order to understand them properly, there is a need for classification. Okay, biological classification is the scientific procedure of arranging organisms into groups on the basis of their similarities and dissimilarities and placing the group in a hierarchy of categories. Okay, you can see in this picture the total living organisms classified into 5 kingdoms. One is Monera, okay, which include eubacteria and archaebacteria, unicellular prokaryotic organisms, the bacteria. Then Protista, eukaryotic unicellular and multicellular organisms. Then fungi, multicellular eukaryotic animalia, multicellular eukaryotic plantae, multicellular eukaryotic organisms. Okay. So, like that there are many scientists who put their ideas, who has developed their hypothesis. Okay, for example, Aristotle 350 BC divided animals into 2 categories. Naima means with red blood and Anaima without red blood. Animals into 2 groups. Aristotle divided 2 categories, Naima and Anaima. Naima means the animals with red blood, Anaima means animals without red blood. And the Linnaeus gave the 2 kingdom system of classification comprising of the kingdoms Plantae and Animalia. Later Haeckel introduced the 3 kingdom classification with the kingdoms Plantae, Protista and Animalia. Copeland gave the 4 kingdom system of classification having the kingdoms Monera, Protista, Plantae and Animalia. Okay. And later R.H. Whittaker 1969, he divided all the organisms into 5 kingdoms, namely Monera, Protista, Plantae, fungi and Animalia. Okay. So, that system still we are following. So, let us proceed children, are you ready? The main reason behind the binomial nomenclature is to group and identify the different types of species which were present on the planet Earth. Binomial nomenclature, it was introduced by Carolus Linnaeus. Carolus Linnaeus is considered as the father of taxonomy. What is taxonomy children? Taxonomy is the branch of science which deals with the study of classification of living organisms. Okay, as we are discussing the lesson biological classification. Okay. So, binomial nomenclature, naming the organism with 2 names, or it is also called scientific naming of organisms. There were around 1 to 2 million types of plants, animals and other 3 to 10 million organisms which were unidentified, means which we do not know, such as bacteria, viruses, birds, insects, etc. There are so many are there. Biological classification helps in quick and easy identification of a newly found organism, which otherwise is a very tedious process, very lengthy process. The organisms of past and interrelationships between the different groups of organisms can be studied properly only with the help of biological classification. This is the advantage. What is the advantage of biological classification? The organisms of past and the interrelationships between the different groups of organisms can be studied properly only with the help of biological classification. Okay. Later, Carolus Linnaeus classified the different organisms based on their structure and discovered the term binomial nomenclature. Okay. So, grouping the organisms based on the similarities and naming them, it is called classification. Okay. The classification system introduced by Linnaeus is binomial nomenclature. It is the method of naming the organisms by their genus and followed by their species. Means according to binomial nomenclature, every organism consists of two names. The first name is the genus and the second name is the species. Linnaeus classified the two kingdoms of classification. What are they? Kingdom Plantae and Kingdom Animalia. But this was not successful. Why? Because as it could not distinguish between. Number one, what are the drawbacks of the Linnaeus system? Eukaryotes and prokaryotes cannot be distinguished in this classification. Unicellular and multicellular cannot be distinguished, and photosynthetic and non-photosynthetic based on the nutrition, it is also not distinguished. Means here the three important factors we are considering. One is the nature of the cell. Prokaryote means primitive cell without nucleus. Eukaryotes means the nucleus, the cells with a well developed nucleus. So, there is no differentiation between them in the Linnaeus classification. Then unicellular and multicellular, okay, there is no differentiation or there is no distinguish between. Next, means though they possess differences, they are included in the same kingdom. So, that is why photosynthetic and non-photosynthetic, here we are considering nutrition. So, both photosynthetic organisms and non-photosynthetic organisms are included in the same group. So, that is why the Linnaeus system of classification is not up to the mark. The biological classification helps in studying wide variety of organisms and interrelationship among different groups. This biological classification system increase as more species are found and classified. Okay. So, discovery of the species. John Ray introduced the term species. What is a species? A species is nothing but the group of organisms belongs to the same population, which are similar in structure and which can interbreed, which can reproduce. So, such a group of organisms come under one species. Okay. In the year 1969, a scientist by name R.H. Whittaker, okay, so he is a very famous scientist. He divided the organisms into 5 kingdoms. What are they? Monera, Protista, fungi, Plantae and Animalia. These are the 5 kingdoms. So, this 5 kingdom system of classification was introduced by R.H. Whittaker. He divided the entire living organisms into 5 kingdoms. The classification of organisms was based. So, what he has taken as a base? He is based on the cell structure, number 1, organization, number 2, mode of nutrition, number 3, reproduction, number 4, phylogenetic relationship. Okay, this is number 5. These 5 factors he has considered. Okay. So, all the living organisms, R.H. Whittaker divided them into Monera, Protista, fungi, Plantae and Animalia. Okay. Next, so how these 5 kingdoms? He based on those 5 characteristics. What are they? Cell type, cell wall, nuclear membrane. So these are all the structures which are inside the cell. Body organization, mode of nutrition and reproduction. Okay. So here, if you consider the first Monera, how the cell type? It is prokaryotic. If you take Protista, it is eukaryotic, means nucleus is well developed, nucleus is present. Remaining all fungi, plantae and Animalia, the cell type is eukaryotic type with well developed nucleus, except Monera. Monera include primitive organisms. Okay. So these 2 major groups of this Monera kingdom include Archaebacteria and Eubacteria. Archaebacteria means ancient bacteria. Eubacteria is further divided into bacteria and cyanobacteria, means about bacteria included in the Monera kingdom. Okay. Then cell wall, if you take the bacteria, non-cellulosic, cellulose is absent. Okay. It is made up of a polysaccharide plus amino acids, whereas in Protista present in some. So for example, Euglenoidea, Euglena is an organism with chloroplast in it and cell wall is present. Okay. So present in some but absent. For example, protozoans, amoeba, so there is no cell wall. Okay. And if you take fungi, there is a cell wall but without cellulose, whereas plantae there is a cell wall with cellulose. In Animalia, cell wall is absent. The next, once again, next characteristic feature, nuclear membrane. Monerans nucleus is very primitive, so that is why they are prokaryotic. So nucleus or nuclear membrane is absent, whereas remaining all Protista, fungi and plantae, nuclear membrane is present. Next, body organization means how the body is formed. It is cellular, means only one cell, or acellular we can also call it as bacteria example, Monera. In Protista it is cellular. In fungi, plantae and Animalia, multicellular. Okay. In fungi, loose tissues are present. In plantae, tissue system and organs are present, whereas in Animalia, tissues, organs, organ systems are present. Okay. Then mode of nutrition. In Monera, different types of modes of nutrition is observed, like autotrophic, chemotrophic or chemosynthetic, and photosynthetic and heterotrophic. In that heterotrophic, saprophytic and parasitic nutrition is observed, whereas in Protista they are autotrophic, photosynthetic and heterotrophic. Okay. Some unicellular protozoans are included in the Protista, they are heterotrophic, whereas fungi they are purely heterotrophic, they are saprophytic and parasitic, 2 types of nutrition in the heterotrophic, and whereas plantae only autotrophic, they are photosynthetic. Okay, they can synthesize their own food, and in the Animalia they are heterotrophic, they show holozoic nutrition and saprozoic nutrition. Okay. Saprophytic means in plants, saprozoic means in animals. Okay, children. Now come to kingdom Monera. Okay. So the kingdom Monera consists of very small and one-celled organisms. Very small and one-celled organisms. They are prokaryotes, which include species such as bacteria, archaebacteria, cyanobacteria and mycoplasma. Bacteria and bacteria-like organisms are included in the kingdom Monera. They are prokaryotic, nucleus is absent, nuclear membrane is absent. They are present almost everywhere around us. They are unicellular organisms with no specific mode of nutrition. Okay. They can undergo different types of nutritions. They are both aerobic and anaerobic, means they can respire oxygen, or without oxygen also they can survive. The presence of cell wall, which are composed of peptidoglycan. Okay. So, cell wall is present, it is made up of a chemical peptidoglycan, and they have naked DNA with the absence of membrane around organelles. So, reproduction is through binary fission. You can take example bacteria. Okay. So, archaebacteria, the two main groups as we have discussed, archaebacteria and eubacteria. If you take archaebacteria, they are a group of most primitive prokaryotes, and archaebacteria are of three major types. They are methanogenic bacteria, halophiles, and thermoacidophiles. Methanogens are obligate anaerobes, means without oxygen they can without or with the presence of, in the presence of oxygen they can live. For example, in biogas plant, these methanogenic bacteria, okay, methanobacterium, methanococcus. And halophiles, if you take, these bacteria can survive in the salt loving bacteriitis, as they are found to live in the environments with very high salt concentration. And thermophilic bacteria or thermoacidophiles, they live in extremely acidic environments, the pH is less than 2, okay, that have extremely high temperature, even up to 110 degrees centigrade temperature also they can survive. They are found in the hot sulfur springs. Which bacteria? Thermoacidophiles. So, bacteria are characterized by peptidoglycan. Peptidoglycan is the substance with which the cell wall is made up of. Okay. The bacterial cell structure that are present external to the cell wall are glycocalyx or capsule. Glycocalyx or capsule is present, this is, okay, or slime layer, it is also called a slime layer. Then flagellum, this is the flagellum. Okay. Fimbriae, fimbriae means pillars, this is the structure, small flagellum like structures. Okay. And the structures internal to the cell wall include plasma membrane or cell membrane, plasma membrane or cell membrane, this is the cell membrane structure. Okay. And next, mesosome, mesosome, ribosomes are present, and mesosomes are also present because they are compensating the mitochondria to produce the energy. The nucleoid, there is no nucleus, plasmid or nucleoid. Okay. This is called nucleoid, which include only chromosome. Okay. Next, the cytoplasm is present. The cytoplasm is the living substance, this is the cytoplasm. Okay. And the bacteria are classified on the basis of shapes, different shapes you have studied already. Okay. Based on staining, based on shape, coccus, bacillus, spirillum, vibrio, and pleuromorphic. And if it based on the staining, gram positive and gram negative bacteria. Okay. Next, kingdom Protista. They are single-celled eukaryotes, single-celled eukaryotes, and mainly aquatic in nature. It includes diatoms, golden algae, euglena, protozoans like amoeba, paramecium, plasmodium, etc. Okay. In this kingdom, the cell walls form two thin overlapping shells. The walls are embedded with silica. The cell walls deposit forms a diatomaceous, which is used for polishing, filtration of oils and syrups, etc. Mostly marine and photosynthetic. Okay, all are unicellular eukaryotic organisms and are placed, they are placed in the Protista, but the boundaries of the kingdom are not well defined. It acts as a connecting link between the prokaryotic kingdom Monera and the complex multicellular kingdom fungi. Okay, complex multicellular kingdoms like fungi, plantae and animalia. Okay, in between the Monera and the fungi, plantae and animalia, these Protista are present. They act like connecting link. The kingdom Protista includes chrysophytes, dinoflagellates, euglenoids, slime molds and also protozoans. Okay. Chrysophytes means diatoms, chrysophytes, green algae, diatoms and golden algae. They are included in the chrysophytes. Most of them are photosynthetic, they are microscopic, and planktonic means they float on the water. Okay. So, you can see this. So, this is the euglena, okay. This is amoeba, paramecium, okay, and the diatoms. Okay. Next, fungi. The fungi are eukaryotic, achlorophyllous, heterotrophic, spore bearing, non-vascular organisms. Cell wall of a fungi is made up of chitin or fungal cellulose. The bodies consist of long thread-like structures which are called hyphae. The network of these thread-like structures are together called mycelium, fungal mycelium. The cell walls are made up of chitin and polysaccharides. Chitin and polysaccharides. Chitin is also called fungal cellulose. Fungal cellulose. The body of fungus consisting of a long slender thread-like structures called hyphae. The network of hyphae is known as a mycelium. In some fungal hyphae, a continuous tube filled with a multi-nucleated cytoplasm, that is called cenocytic hyphae, means more than one nucleus is present. Okay. In others, they have septae or cross walls in their hyphae. Such type of hyphae are called as septate hyphae. Okay. The mode of nutrition, if you take in fungi, they are saprophytes or parasites, and some of them are symbionts also. This kingdom of fungi also includes lichens, mycorrhiza, aspergillus, etc. Reproduction is mainly through sexual, asexual and vegetative reproduction. Is see different types of fungi here, the mushroom, okay, yeast, the different types of fungi growing on the different substratum. Okay. Next, further we are going to discuss in the next class. So, before going to end this fungi, the fungi they reproduce by three methods actually: vegetative, asexual and sexual methods. The vegetative reproduction takes place in the by fragmentation, budding and fission. Okay. So, I hope you understood, children. Keep reading the book. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.